Hey grade 12s, it's Mr. Boyle. Hopefully everybody's doing okay. I'm going to try to do a couple of videos on sort of the more important parts for this Unit 2 on DNA and bioethics. The first one's on DNA replication. I'm going to try and keep them to a couple minutes. So on DNA replication, this happens during interphase of mitosis. We can't see it happening. Remember, it looks like a jumbled ball of spaghetti during interphase, but through many experiments, this is what we figured out happens. It creates a whole new set of chromosomes so that when we go from one cell to two cells when it splits, we have identical sets of DNA or chromosomes so that the cells function in the way that the parent cell did. Now, during this process, the hydrogen bonds between the A's and T's and the G's and C's, remember those are purines and pyrimidines that are joined together by a weak hydrogen bond, they are broken by an enzyme called DNA polymerase. Now, this happens at many locations along each chromosome during interphase. That makes it happen faster. We call that the replication fork. So the DNA literally unzips. They call it the unzipping process. And that exposes these bases. Those bases act as a template for new bases to come in. These bases come in and bind accordingly at the correct spots. All right, all along to help fill up and make two identical strands of DNA. You can see here we call this semi-conservative replication because the DNA on the outside of the ladder, we would call that old DNA that acts as a template, and then the new bases that come in and bind, and they bind on both sides, is the new DNA. So when we say semi-conservative, half is conserved, half is new, and what we end up with is two identical DNA ladders, all right? And then when that cell splits, you get one goes to one cell, one goes to the other. And then that cell can function because it has a complete set of chromosomes with identical DNA. Now, if this process, there is some problems in it, that could be a type of mutation. Take care.